All right, we're going to go get started. I'm incredibly honored to invite three amazing scientists for this panel today. We have Catharine Aquino, who is at the Functional Genomics Lab in Zurich. Kaitlyn Petentler, who is a scientist at Sowers Institute. Anna Els, who runs the Functional Innovation Lab at Fred Hutch. We also have a presentation by our sponsor, 10X. This meeting cannot be done without the amazing support from our vendors. So if you haven't stopped by the exhibit hall and haven't visited the exhibitors, please do so. And I'm so glad 10X is a sponsor for this session because it's very appropriate. So we'll get started with Joe, and then we will go in the order that they're seated in. And I want the end of this session to be a discussion focus. So please feel free to ask questions. I hope we can have a very productive session.
I'm all queued up, so I'm ready to go. Thank you guys so much for coming out, showing an interest in some of the current methods in single-cell technologies and cores. Thank you to all you guys for offering us this opportunity to share with everybody as well. So today I'm going to have the opportunity to talk to you guys about how at 10X we're trying to continually innovate, continually kind of expand on our products to make sure that they're not just for the highest of funded labs or the highest of throughput projects, but enabling those single-cell multi-omic outputs for kind of any project scale. So if you're maybe new to the single-cell world or you're thinking about diving in, at 10X we try to offer a single-cell readout in as many ways as possible and accessing as many different sample types as possible. So it may be that you're working with fixed samples or FFPE, or maybe you have some fresh cells, tissue, nuclei. And so we have a really great build out of different protocols and options to kind of access any type of sample. And this is from human and mouse species, which may be a majority of what people are working with, kind of into the most interesting species that you might be working with in your lab. And we offer these readouts from gene expression, which is kind of like the basis of what most people would think about for 10X as single-cell RNA sequencing. But as we've gone along, we've added on additional capabilities, whether that be intracellular or cell surface proteins, the expansion into TCR and BCR receptors for VDJ information, kind of adding on the flair of being able to detect guide RNAs in conjunction with single-cell gene expression so that we can directly track what happens to from a perturbation. And then kind of what I'm going to jump into next is the combination of single-cell gene expression with chromatin accessibility through an ATAC-seq assay. So previously we had our next-gen multiome assay, which was enabling single-cell gene expression and ATAC from the same cell, kind of overcoming the bioinformatic challenge of linking gene expression with chromatin accessibility. If any of you have run that assay, it was a very powerful assay, but also is sometimes a challenge to get the highest level of gene expression, or maybe a challenge to get enough nuclei, enough high-quality nuclei to go into that assay. And so we're releasing our new GEMX Epi Multiome assay. It is currently in pre-order, and it will be shipping very soon. And what you can expect from this new version of the Multiome assay is about a 50% increase in gene expression compared to the previous version, as well as an improved reproducibility for the ATAC side. So with that, we've updated a few steps on our transposition step within our assay that is going to really help boost that ATAC reproducibility. The other things, if you followed along, we've moved from NextGem to our GEMX chip architecture, and that allows us to boost that throughput from about 10,000 nuclei per channel to about 20,000 nuclei per channel. That comes along with a reduced multiplet rate, as well as an increased capture efficiency. So more of what you put onto that lane is going to get captured at the end. And kind of the cherry on top of all of this is all of this comes at about 20% cost reduction compared to the previous version of the assay. So you're getting a better assay for way less. On top of the chemistry kind of being updated and more sensitive, we're also updating some of our sample prep guidance. So we do have a new nuclei isolation kit with an associated user guide, as well as we updated our guidance for single-cell suspensions, as well as complex tissue nuclei isolation. So that whole slide was talking about sensitivity. Some of you are like, great, show me the data, and then let me go play with it, and I'll tell you, how right you guys actually are. And so it's always good to start from the 10X side. So this is what we were able to generate from mouse lung as well as mouse kidney. We also have a PBMC data set that will be released at the ship of the product. What we can kind of see in green is the GEMX multiome versus the next-gen multiome. Green is very much a pretty significant improvement on the genes per cell as well as total transcripts or UMIs per cell in both cases, right? And then on the attack side, right, the improvements are more akin because the attack was doing pretty good. It's the assay itself. It tends to be a little bit more reproducible, a little bit less error prone when it comes to the sample prep side. So some of the things on the sample prep side, why I keep on saying why it might be more reproducible or core enabled. One of the biggest changes that you might notice is we changed the nuclei input volume from 5 microliters to a max of 15 microliters. So that means you're not going to be required to get as concentrated of a nuclei input to go into the assay. So maybe less clumping going into that chip or the ability to just do more nuclei. We also updated our guidance on transposition time. So that way from cell suspensions, we're doing about 30 minutes, whereas from tissues, we're recommending about 60 minutes of transposition time. I think the biggest update, which I think is driving some of this increased gene expression sensitivity. So one, because we're doing more nuclei, we're going to need more nuclei input, but we also recognize that some of the challenges with this assay is you're working with precious samples oftentimes, and you can't predict if there's something up with that sample or if for some reason there is an unexpected chip failure. And so this ability to do a scalable bulk transposition of up to 75,000 nuclei means that you're not going to use all those nuclei to go straight into a lane. And so having a little bit extra, as well as this runtime dropping from 16 minutes to 6 minutes, means that if for some reason the worst happens and there's a failure on the run, you have plenty of nuclei extra to load right away, and they shouldn't be compromised. We also added in a step of post-transposition wash, so that way we can clean up some of those nuclei that might have been compromised during transposition, as well as a post-wash count, so that way you get a more accurate prediction of what you're actually loading onto the chip. So you're getting, there's less likelihood to really underload or really overload, and maybe under-sequence at the end of the day. Switching gears a little bit, you've probably heard a lot about GEMX Flex Apex if you went to our pre-event lunch session, or if you went to our breakfast session yesterday. But I kind of just want to reiterate some of the values of this new assay format. And then, again, reiterate how we really want to be working with the cores to make sure that this is something that, regardless of your scale of your core right now, that you can work with and utilize to the full extent. So with the GEMX Flex assay, Flex has been out for quite a while. It's our probe-based assay that enables single-cell gene expression detection from fixed samples as well as FFPE. The way that we updated going from the previous version to Flex Apex is we increased the throughput from about 320,000 cells per lane to about 1 million cells per lane. We also really opened up the flexibility of the assay from stopping you to be, we used to kind of limit it as a 4-plex or a 16-plex, and that was kind of it, and not all projects fit those numbers. And so we've opened it up where any plex can be used in this kit, and it is very modular, so you can do anything from 2-plex to 384-plex. And part of that is done by, I'll show you on the next slide, our probe-based chemistry. We separated the labeling with the probes from the sample ID, so that way it doesn't matter what you're starting with. You can always multiplex to whatever extent you need. The reason why we're, again, so excited about the Flex assay is one, the probe-based chemistry does allow you to kind of get improved sensitivity for those lower expressing transcripts as well as lower quality samples. So it kind of opens up the realm to not have to have a perfect fresh sample to go into single cell and get good data. It also opens up the possibility for functional genomic screens using like custom probe spike-ins so that we can detect guide RNAs as well. But the sample type compatibility also is what we're excited about. You might already have fresh tissue and you want to go into this assay. That's great. It opens up your possibilities for larger single cell experiments. You might have already frozen your tissue because you might have thought about going into a nuclei isolation. You can still access the Flex assay from that as well. And I think the most exciting part is the FFPE, right? There's thousands and thousands and thousands of FFPE blocks that like what are we able to do with those now? It might be an H&E image or some type of IF or IHC readout, but that might be all you thought you could get out of that block. And so being able to access single cell gene expression information from FFPE blocks, you don't actually have to generate a sample. You just have to go talk to your biobank and get some of that sample to get a data set. As we increase the Flex and increase the ability for you to really dream bigger in single cell experiments, we need to also help you out on the sample prep side. And so with the Flex Apex launch, we also launched this new xylene-free plate-based dissociation method. And so we previously used a xylene-based deparafinization, and it was tube-based, and it was a little bit clunky. I don't want to talk bad on our method, but it wasn't the highest throughput of methods. It took a lot of effort and optimization, and so it really slowed down the uptake of this assay. And so this plate-based method not only is less toxic with the xylene-free base, but also it is already in a 96-well format, which means that format or the samples that you prep here can go directly into the Flex assay without having to do any extra handling. Another kind of exciting update, which is very much more recent, is maybe some of the challenges on the Flex assay has been there's a lot of counting, we got to fix, we got to do the hybridization, and so we need more hands to do that, at least at the scale that we want to get to. And so with the Integra Assist Plus, there's a recent method that's been released from the Integra Assist side that has been validated in customer hands, at least through I believe a customer advisor. And this has been able to help semi-automate fixation, hybridization, normalization steps, and then parts of the library construction method. So if that's that's great for you, I maybe already have that, or that's what I was thinking about, maybe adding into the lab. I would encourage you to talk to Integra and maybe talk to their support team and see if it's something that you could try out in your group. So again, why do we harp on Flex constantly, the last couple of days? And I think it's because we start to see this potential of what this probe-based chemistry and what this scale chemistry is able to allow us to do. So this is just two recent examples that came out early this year in bioRxiv. One is ProperSeq from UCSF, where they were able to employ the probe-based approach for a wide-scale perturbation screen. And they were able to really significantly cut cost and increase sensitivity and gene coverage by using this probe-based approach versus our 5' or 3' CRISPR-based approach. And then VI PerturBSeq, which is coming out of the New York Genome Center, Raul Satij's lab, they kind of took it an extra step. They used the new Flex Apex chemistry, and they did a very large screen, right? So 57,000 perturbations. But this method is kind of cool because you can kind of select which of your perturbations are most interesting to you, which is the VIP part of that seq there. And so the way that they got around kind of just actually targeting the guide RNA is they created a barcode library where you could actually detect all your different perturbations with only 500 custom probe spike-ins versus the standard of every guide gets its own spike-in. And then another more recent update, maybe on the industry side, is Perturb AI. They recently used Flex Apex to do a in vivo whole brain perturbation screen and got an atlas of about 8 million cells. So that might not be where you guys are at, right? But it's what the chemistry is starting to enable. So I want to make sure I'm conscious of time, so I'll jump through real quick and say, you might have seen this in our breakfast session before, but those studies are great. Some of them are pharma funded. You guys might not be able to be running projects that are pharma funded where you can just do millions and millions of cells, but that's where the chemistry is allowing you to start thinking about getting to, right? But you might not be there yet. You might not be able to take advantage of that 384-plex kit, which really gets you down on the cost, which means you can't take advantage of the pricing, which means you can't get the projects that are hoping to get there, and it kind of gets you in that cycle where I would love to get there and price it that way, but I really just can't feasibly get to a 384 kit. And so that's where we really want to start to come in, and Jason Kim talked about this yesterday, but we want to start partnering with you guys. And it's going to look different for every single core and every single specific use case. But we want to try to meet you guys where you're at. We want to help you get to the scale that makes the most sense for your group and kind of long-term sustainability. So it might be in enabling, getting some of those new to single-cell customers a little bit farther along, a little bit earlier. It might be talking to some people that may be coming to you for bulk right now and asking them before, are you going to do single-cell later on? Is this just something to maybe push the project along enough to validate doing single-cell? Maybe it's worth us talking there too. We can help on driving demand, but I think the biggest thing is these last two sections. How can we help to make sure that when the sample finally gets in your hands, it's going to be successful at least as highly as possible? And then once it is successful, how do we get that user to kind of get past that data mountain, figure out what they learned from their sample, and maybe come back for additional business? So what I've talked about today is kind of the latest in the GEMX Flex Apex. So if you didn't hear about that before, now you should be up to date. We want to make sure that we're partnering with you guys to make sure that that is a possibility. And then I updated you guys on the latest GEMX Epi Multiome. But there's still more to come, right? And many of you guys came up to the booth already and asked us, what is this impossible thing? I would love to know as well, but if you're just as curious as I am, I would encourage you guys to come out to this 418 event. You can scan the QR code or stop by our booth. We have some flyers for it. Whether it's on the single cell or spatial side, I think it's going to be exciting. So I'm ready to figure out what it is as well. Thank you guys so much for your time, and I'll go ahead and pass it off to the rest of the session here.
Uh-oh. I actually wore heels for this, but it's not enough, right? You still can't see me. Um, what do we do? Okay. Yes. Doesn't work. Ah, okay. Okay. Yeah, so a few months ago, Anoja asked me to give a talk about the challenges that we're facing with single cell services. And then she said, like, yeah, just talk about your challenges and, and your solutions. And I said, what do you mean? Can you be more specific? Because when I think about it, single cell is, we have been like, we implemented single cell like 2016, 2017, like almost 10 years. And so basically, when we were doing only bulk, at this point, 10 years bulk, we just changed from TrueSeq maybe 2 years ago. So what I mean is it's already very mature and the bulk. But with single cell, we're still basically constantly trying to improve the service and trying to adopt the new methods. So then I was inspired that I will talk to you today about the epic saga of single cell services at the Functional Genomics Center. Yeah. So we are a core facility of two universities, which is the ETH Zurich and the University of Zurich. And so we have, genomics, proteomics, metabolomics, and bioinformatics in one roof. And the genomics team, so we have NGS and long reads, but at the same time, we're also the single cell facility and the spatial facility. And all this is bound together by our genome informatics group. And we have a lot of sequencers, and we're also running a lot of boxes of single cell. We are offering all the vendors for single cell. And yeah, so if you have seen my slides, because I was also giving the talk yesterday, sorry, but well, but as a core facility, you know that we are, like a Mission Impossible team, because we have to be state of the art, standard, customizable, fast, reliable, affordable, and yeah, I think the affordable is also very important. So anyway, so the epic story of single cell services at FGC set. Please thank Gemini for a very nice photo. And yeah, so basically, I want to. We have different, how you say, eras of single cell sequencing in the center, and I will tell you all about these different books, because it's like an epic story. And then we also have side stories that I will go through later. So I want to say, like, in the beginning, like, I would call it the dark ages. So basically, everyone is trying just to implement single cell. When we were attending the 10x user meetings, I think I was probably the only core facility there, and most of the user meetings in this period were research groups. And so basically, what was the problem then was the lack of expertise. So basically, people have NGS expertise or cell biology expertise, but no one really have both expertise. To prove a point, we actually believed, because I was running the single cell myself, and I actually believe the cell concentration the users tell me that it is. Can you imagine? Yeah, and so in these 3 years we managed to form the expertise, but unfortunately, as soon as these people are formed, they just go to the companies because they are paid better, and so we always have to how you say, build expertise and build the experts, and we even lose how you say, people to 10x, but it's okay. I'm proud. Yeah. So, but the decision before was easy. So either you do droplet or place based, and then at that time it's already a problem that the samples are always coming late. The samples after fax they come like at 9 p.m. on Friday, and then you're like, I have a life, right? So, but yeah, good luck. Anyway, so how we solved it is we solved it by actually training more people, so we're not dependent on one person for the services. And then we tried to see, do we do droplet or plate based or both, because at the time anyway when you want to go deep, then you do droplet plate based, and then if you want to do a lot of cells, then you do droplet. That's kind of an easier decision. Yeah, and then so we also had to how you say, talk more with the fax facility, because we were existing like independently before. No one really knew what the other one was doing. I mean, maybe still a bit now, but at least they know what we need from them. So 2021 the next age, or the next book, is the what I call the age of enlightenment. So in this period, basically the problem most we knew suddenly. Okay, now we know what we're doing, more or less. But then the 10x kit, started evolving the versions. You have, I cannot keep up, like version 1, version 2, version 3, and then you have a lot of kits in the stock room because you bought a lot the month before, and then suddenly there's a new kit. Yeah, so you know how that works. And then, of course, now there's a lot of paper, so users become more demanding, and then they expect more from you. They expect, like, that it will solve all their problems if they do single cell. But of course, it's not like that, and so you have to manage expectations. Say like, oh, if you're sorry, that's my solution. So we have to do a lot of expectation management. So basically, if we have to tell the user, if your samples are dead, there is no way we can do anything about it. Also, of course, samples still always come late, right? So it always still come at 9 p.m. on a Friday, and yeah. So what we try to do is we try to schedule the projects, and then so we know when the samples are coming in. So we try to like, just even from the start of the experiment, when they're doing the mice study or so, we know when it will come, so that at least we can plan our life around these things. So we also implemented the single cell in our limb system. So everything is transparent because samples come in, and then so basically, and then the user, they've been sorting for since 2 a.m. and then it's 9 p.m. and then they're like, I just want to get it done, and then you're like, oh, but your cells are dead. And then they say, but I really need to finish my PhD. But then the problem is, if it doesn't work, it's our fault, and then they don't want to pay, right? So yeah, so what we try to do is, we have like in our limb system, we have a common functionality, and then it's written there, the user wants to proceed. And then this is also basically their PIs also see this message, and so everyone is happy, and we kind of are protected. Okay, so the present age, the what we 2004 to now. So the challenges are different, because now you have a lot of different vendors, a lot of different technologies, and so basically, like, now it was exciting before, because before it was only 10x, and then suddenly now there's a lot. You have Pars, you have Illumina, you have everything, and then but now it's like, okay, what is that all good for? Is that really necessary? So we have to keep up with the new technologies, and still, of course, we have to always update our users, educate our users. And so now what we try to do is, we do actually continuous benchmarking. So we try all the kits and to see what it's good for, and then we try to see, like, what's problem does it actually solve, that we have to implement it. And then one problem is now with the Pars or with the Flex, you can do like a lot of samples, so you have to you have to figure out how to count the cells, how to QC the cells, because yeah, you can do 384 samples, but your Luna is one by one, so good luck, right? Yeah. So anyway, still samples are still arriving late. It always is like this. So what we know now, so what we think we know now, that it's actually always have been and always still is a sample and workflow problem because the cells still need to be alive and the cells and so on. So I think the chemistries are very similar. So you get like 50 more genes, 50 more transcripts or something. So all the chemistries work. But what's really important is that the protocols are streamlined. So for example, here in this photo, this is like an 8:30 p.m. In the evening. And then my team was joking that use this kit if you never want to go home. So do you think they will offer this kit ever again? No way. You know why? Why do they need to do that? Well, okay, they do it. But because they're good from the bottom of their hearts. Because some cells it needs certain kit because it's less fragile cells, for example. So they actually know, okay, now I'm not going home. Yeah. So basically, I really think that single cell it's not necessarily a chemistry problem, but it's more a workflow problem. And so, now we're going to the side story. What our current problem is, this is what we call the workflow of death, because basically you thaw the cells and then you wash and then you process these cells, and at the end of it, it's dead. Because the way you process single cell, it's long and fragile and multi-step, and then small mistakes lead to a failure. And then this leads to slow viability and counts. Cells are stressed. It takes ages. And then my team is stressed. They get they're not viable. They're stressed and they age as well. So the solution can be, I always think this is the hope, is fixation. Because fixation, it decouples now the sample collection and the workflow. So nobody has to stay late, right? And then so my team can go home on time, they will be less stressed, and they can stay young as well. So it looks like the solution at this point. But unfortunately, it requires very high cell input, and the recovery is very low. And I think the problem as well, so for example, when we have some projects with PARS, the user fixes the cells, but they don't QC it. And so after sequencing, they realize, oops, it was dead actually when I fixed it. So it still requires a QC step like before the fixation, or to figure out if you what kind of cells you actually fixed. So this is just to show you like the recovery of when you fix cells. So the recovery is still quite low, and because you need a lot of these washing steps. So with some friends from Vienna, so Vienna Biocenter, we are trying to benchmark some fixation methods to see if it will work for everything. So the hope is, we find something that can work for different chemistries. Yeah, so we are working on that. And then another thing that we are trying to see what we can do is high throughput QC. So right now we're just having a Luna that can count 8 samples at a time, and yeah, so it's still not the most optimal solution, but at least it's 8 samples at a time. And then of course the big problem is I think there's a concurrent session doing the same thing, but it really I feel it. I think it's really one of the problems for a core that if it doesn't work, I don't want to pay. And so we really have to do a lot of expectation management, communication, define QC checkpoints, and then if they want to proceed, then I think it should be clear for everyone that they still need to pay. Yeah, and continuous benchmarking. I will not go through the results here, but we are, so for example, here we benchmarked the Illumina single cell versus 10x genomics. I'm not going to go through it, don't worry. But at the end, so long story short, it still managed to 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 get the to get it done, to get the job done, right? Yeah. So these are the current technologies we're offering. So basically, we're offering everything anyway, and yeah, I'm quite proud of this. So you have to see. So this is like the map, the landscape of the single cell vendors that we are currently offering. So as you can see, 10x is the green, BD, PARS, and yeah. So it also means something, the islands, but but I see people from the company, so I'm not going to explain it. Okay, so for automation, we're also quite fond of the Integra Site Assist Plus. And so we are also doing this modular automation with Integra. So now we are running it with Parse, and that works quite nicely. Yeah, full disclosure, I know also people from Integra, so I'm biased. Yeah, so the strategy today is we run multiple vendors. So, and then we do continuous technology improvement. We try to match the kit to the content, to the project. We control the workflow and not only the technology. And then what is really, really, really important is a foolproof LIMS documentation. And that is the ending. So basically, this saga will be continued forever and ever, I think, unless someone solve the real issue, which is the workflow. Thank you very much.
All right, I might also need a little assistance. Get a little help with this real quick. And then just to get into the right mode. All right, perfect. All right, hi everyone. My name is Katie, and I'm a member of the Sequencing and Discovery Genomics Core at Stowers Institute. Today I'm going to give a little bit of an overview on how we handle single cell with non-model organisms, and also talk a little bit about a benchmarking study that we did to bring in new technologies to work with these type of assays. All right, now Stowers is in Kansas City, Missouri. Missouri is an important distinction. And we are a basic biomedical research facility that has about 500 members, spanning between our research labs, our core facilities, and our grad school. And so we see several different areas of research being conducted at the Institute. And to really study these, we have several different models that come through our lab. Some are pretty standard, such as mouse or zebrafish, and others that are quite unique. We've had apple snail, nematostella, and coral. So this is our team. You might recognize some familiar faces. We have several primary services within our lab, such as Sanger sequencing, plasmid preps, and we have a few different flavors of sequencing in-house for long read and short read. And then we have a variety of different next generation sequencing workflows, which we'll talk about today with single cell. Now, when working with non-model organisms, it's important to take into consideration several different factors to pick which assay will be best to use. For sample collection, there can be limitations. Some of our researchers are traveling around the world to collect these types of samples, so they may not always have the opportunity to get more if we need to do a lot of optimization. Sometimes the researchers do have experience with the organism that they're working with, but if it's something new, or if it is a grad student who they've been thrown a really unique organism, they may need a lot of optimization to go into working with that organism before we go into single cell. Now, cell size can be a limitation, especially depending on the organism. Maybe they have a pretty large cell size that they're working with or really wanting to explore. So depending on the downstream assay, this could really be a limiting factor. There also might be some upstream constraints just to maintain the sample integrity that could affect whichever assay is used. For example, we have some marine organisms at our institutes, and they require specific needs to maintain their cell integrity. And so this is something that we definitely have to keep in mind when looking at assays to use. One other consideration that I kind of want to point out is something that we actually had come up recently, which is a well-annotated genome. For some of these more unique systems, there may not be the available, like, widely studied annotation that we would expect with a model organism. And so they may have to create this or curate this themselves, which could lead to a little bit of delay with analysis downstream. All right, so within our single cell service, we do have a few different options to offer our researchers that we've been using for several years. We have a low throughput method with SmartSeq by Takaro Biosciences. And this is a really nice plate-based format so that if our researcher is looking at rare cell types or have very limited cell numbers, they can fact sort individual cells directly into the wells of the plate. And we do offer a way to miniaturize this to be cost-effective for our researchers as well. More often than not, we do see our researchers doing a high throughput method, and so we typically offer 10x genomics in several varieties of their assays, and this allows us to use microfluidics to capture tens of thousands of cells. And you can see just the examples of our commonly used assays here. Now, for all projects, but particularly important when we're working with non-model organisms or new to our institute organisms, is to conduct a QC pilot ahead of processing. This allows our researchers to get an idea of the dissociation that they'll need to do, look at the different parameters for fact sorting, and if they aren't using fact sorting and they're noticing debris, this would be a great chance to kind of evaluate if that is needed. This also gives us the chance to look at the cell numbers and nuclei quality if they're choosing to do a nuclei version, and to assess the viability. And so here I just have a few brief images of what good QC should look like. You'll have a nice singulated cell suspension. Typically, we are looking at this on our Luna cell counter that we have in our lab using AOPI dye. And if a lab is interested in using nuclei, we encourage that they go to our microscopy core to really look at the membrane. All right, so as we continue to expand the amount of model organisms that we have at our institute, we are always looking to bring in new assays that we can offer to our researchers, just to give them a little bit of variety. But when we are bringing in these new technologies, there are some key considerations that we want to make sure we are keeping in mind. Will the technology work with the variety of unique organisms that we have at the institute? Will this meet a current need of one of our research labs or a research lab that may be incoming? And is the technology going to bridge a gap that we may currently have in some of our offerings? All right, so we wanted to conduct a comparative study to look at a few different tests or assays that would be coming into our lab. And so you can see the ones that we were looking at here. We really wanted to explore the new genomics protocol as it came out because this is something that our researchers are really familiar with. They've been using the previous chemistry for years, so we're excited to see the new and improved capture rates and sensitivity. But along with that, we did want to see if we could bring in a type of fixation model. So we have our parse and scale type of assays that we wanted to test to see if these would hold up well. And this is using a combinatorial indexing approach, and this can be scaled up. Now, we had first conducted this initial testing with just these three types of assays, but once Illumina kind of acquired the Fluent Bioscience PIP-seq protocol and assay, we were interested to see how this was going to work. We had previously tested this many years ago when this type of technology first came out, so we're excited to see the new improvements and if this would be something that we could offer. All right, so because we were wanting to do a comparative study to really investigate how the assays would compare in terms of chemistry and ease of use, we first wanted to use a barnyard mix because as long as it could work well here, this would be something that we would want to test further as soon as we get new model organisms. But this barnyard mix is kind of a standard that we use for a lot of our benchmarking testing with a human and mouse 1-to-1 ratio. And so for our initial assays with genomics, parse, and scale, we did take the same aliquot from the same cell suspension. But with Illumina, we just used the same type of mix. And you can see here what the QC should look like. Again, we had that high viability. It looked very nice in a nice single cell suspension. And so we took the fresh cells directly into our GEMX and Illumina assays, and then we fixed aliquots with our parse and scale. And then we just looked at them again in QC right after the fixation to make sure that with our AOPI, these were kind of staining red, and so they would appear dead. So we didn't move forward with processing through the protocol for each of these individual assays and sequenced. And so we wanted to first look at the multiplet rate for each of these assays. So as you can see with the GEMX and scale, this is looking at what is typically advertised for each of these respective companies. But the parse sample had a much higher multiplet rate than we had expected. We went back to our QC. We had worked very closely with our FAS, and they offered a few guidance for adding in a few upfront reagents, such as DNase treatment and RNase inhibitor, to the fixation portion of the protocol to really help alleviate the clumping and multiplets that we were seeing. And so when we repeated the assay, we were in line with what we were expecting. So here's just a quick bar chart to look at it in a different way. And in the next slide, I will only include the results from the repeated parse sample. But what about Illumina? We wanted to look at this kind of side by side. And as you can see, we had a really nice low multiplet rate that was in line with our other assays that we were testing. So the last two kind of QC passes that we wanted to look at for this benchmark study was the capture efficiency. And we were really happy to see that all of these assays were having a high capture rate with over 98%. We next wanted to look at the median gene and UMIs per cell. And these were pretty comparable, but we did see a little bit less as we were using the parse and the scale and the median UMIs. But overall, we were happy with this. This looked comparable enough that we were able to offer this to our researchers for new projects that would come our way. All right, now this is a very busy slide. We don't need to look at all of these in detail, but feel free to take a picture if you would like. But I wanted to highlight this as this is a nice way that we typically summarize a comparison assay like this when we're looking at different type of technologies. And we use this when we're talking with researchers as we get new projects coming our way to really see what would be best for their specific project and their organism that they're working with. And so we're really happy with, we're really fortunate that right after the study actually completed, we had a researcher who was interested in using a non-model organism that we hadn't worked with before. So we looked at this and were able to figure out which would be best for her project. And that was a wallaby sample. Now this is definitely something different. This was new for us, and the researcher had actually started working with this organism within the last year. They had to go all the way to Australia to collect the samples. And so there were some upfront challenges with the sample type in particular, since it was going to be limited samples. We suspected low cell numbers, and there was unknown RNA content. But our researcher had worked with our tissue culture core to immortalize these cells, but they weren't sure what exactly the cell ratio was within this type of sample. So that's why they were wanting to do single cell sequencing in the first place. With their sample, they knew that they wanted fresh. They had been working with 10x in the past, and so they had came to us originally wanting to use the 10x GEMX, but they were gracious enough to let us also test this side by side with our Illumina assay. This allowed us to kind of scale up from our initial testing with Illumina just to see how this would look outside of a barnyard sample. And you can see here we had really nice QC going into this. We used the same aliquot and kind of split them between the two assays. All right, so just to look at a little bit of a QC comparison, our researcher is actively looking into this and more in depth of the wallaby cell line for both of these assays. But from our standpoint, when we're looking at a first pass QC, we did notice that the capture efficiency was a little bit lower for both of the assays, with GEMX being a little bit higher, but the overall median genes and UMI were pretty comparable, though we did note a bit of a distribution, or a longer distribution with the Illumina sample. All right, so just to wrap up and give a overall summary of what I've talked to you guys about today, the non-model organisms that we work with are really a unique way to study a biological question, but there are careful considerations that we do have to take into account when using these for single-cell assays. Proper QC and optimization are crucial, especially when we're working with these non-model organisms. And when we are bringing in new technologies, doing a side-by-side comparison such as this, or such as what I have shown, is a really great way to really evaluate the different assays that we could offer. And with our testing comparison, these were comparable enough that we were able to offer them as other alternatives for what we're using at our institute. With that, thank you.
Hello, everybody, and thank you, Anoja, for inviting me to come speak today. Well, here's the word of the morning. Obviously, it's benchmarking. Everybody does benchmarking with single cell. So I'll be sharing a little bit about what we've been up to. And yeah, I'm at Fred Hutch, and I run the innovation lab. So I'm not a traditional core. I always have to sort of clarify that. I work within our Immunotherapy Integrated Research Center. We started the lab in 2020 to specifically onboard and test and compare different single cell and spatial technologies with the goal of getting these platforms into the researchers' hands faster so that we're developing and gaining more insight about immunotherapy. And that's really what our mandate is. So we work a lot with sample prep. Say we'll test different ways of processing tissue samples, fixing, non-fixing. We do a lot with this new DSP fixation. We obviously do single cell benchmarking. We have a great data scientist and bioinformatics specialist who has developed a cell segmentation method called ProSeg, in addition to some other types of tools. And then we collaborate a lot with different groups because we're always looking at how to kind of push the envelope a little bit on how we're doing analysis and integrating different types of data sets. And so there'll be a couple samples that I kind of show and share with you in the later part of my talk that we've just used over and over on different platforms, and it's a sense of benchmarking. So just a really brief background on benchmarking. As Catherine pointed out, she's been doing this since 2016. I think before our lab started, we had single cell around 2017 or 18. My tech was one of the earliest ones to do it. But people have been benchmarking this since its inception. And they've gone from both the wet lab, the workflow side, the metrics, to different types of analysis. And so, of course, we did this as well. The most recent one, in addition to what Kaitlyn just shared, which I think is really important on different species, is that you're looking at whether or not you're relying on the platform's pipeline or looking at some other agnostic type of analysis. So this was a study just recently out of the Duke Genomics core. And then Lou, if everybody knows Luciano, he's sort of drafting this benchmarking playbook because it's really trying to get us all to be thinking about how we're doing this. And we see a lot of consistencies and asking what are the types of samples? How many replicates? Are we having different labs do this? What's our biological question? And I think these are all really important things that we should be keeping in mind. So just a little brief kind of background on the benchmarking study that we did. It was basically right when GemX came out. And so we had a lot of users that were doing the next gem. So we were asking ourselves, just like Joe shows you the data up here, you've gone to these other user group meetings. You want to trust them, but you really want to see it in the user's hands. You want to know what am I capable of doing? And so we basically tested this GemX against its original, along with some other platforms that we were really interested in because obviously we were thinking about throughput even back then before this Apex thing came out. And so we did it in a kind of different way. We did it as a 2-benchmark approach where we tested 10 different kits. Specifically, 3 of those were kits that would target a TCR library because we're immunotherapy. Everybody's interested in T cells. They want a TCR library. And we broke this down into like looking at reproducibility with replicates in our first benchmark, and then looking at kind of more of a variety of different donor samples in our second. Obviously, same metrics, recovery, scalability. Thinking about workflow, we were trying to understand the practicality of either an individual user doing this versus what it looks like doing it in a core. Cost always comes up. What are you getting for the money you're putting in? And for us, cell composition was also really important, as well as your sensitivity. And this just got published in January. So in our case, very similar to some of the data you've seen earlier, we were looking at the recovery metrics. So it was supposedly going up from 50% to 70%. And we did see that. We saw a big improvement in the GemX. One thing I want to note here, too, is that this was all done on fresh cells, except for where the kit required it to be fixed. And for Parse, they have their fixation. Scale has a DSP fixation. For Fluent 4 here, we tried their early version, and we compared it with their newer kit, which I'm calling Fluent, but it's now Illumina. And so we did fresh for that one. And quite honestly, what we're looking at is, for the little dash black line, how well are they performing based on kind of what the expectation is of what they're predicting you'll get. And on the right, we really like understanding like how much information are you getting for the amount that you're sequencing. And for this one, we borrowed the RD50 score that the DeSimone benchmarking paper created from their previous benchmark. And so the idea of the RD50 is that it's looking at if you have a higher value for your RD50, it means you're going to get more information by sequencing more deeply. And so here we see that even though Flex always does like really great, it's efficient, you only need your 10,000 reads per cell, we saw like that GemX had a really high recovery on your median genes per cell, but that if you kept sequencing more deeply, you would actually get more information. And the thing with Parse and Scale, what's interesting here is it looks like you're getting like the lowest recovery, and I just want to like kind of point out that that's really based on like the limitations of what the, or not the limitations, but the expectations of what you're putting in. They are telling you are going to put this much in order to get this many evenly back across your samples in the plate. And so it looks like it's really lower, but that's like a percentage based on the total number of cells that you're putting in. So in essence, you have to kind of know that, like going in, you need a lot more cells because there is that loss through moving things through the plates. And for us, obviously, that was a big one. Resolving the biological signal was really important. So we looked at things like, is there significant enrichment for particular pathways? And for all of the kits, we saw significant enrichment between monocytes and B cells for the monocyte pathway. And then similarly, we saw significant enrichment for the TCR pathway when we were looking at B cells and T cells. However, we only saw a significant enrichment for the monocyte pathway between T cells and monocytes when looking at Flex and GemX, and that was important to us. And that was, that was our benchmarking study, and it was a lot, and this is what happens where essentially you do all of that work, and then the next kit has come out, and you are on to testing something else, and you hope that what you've put out there was of some value to the labs, and I think that's what is worth it. So I'm going to share with you what we've been up to now recently, which is the Apex, and we have 3 studies kind of going on. And for the first one, this started actually a couple months ago because it takes some time. We have whole blood that we've been fixing over the course of a couple of months from a patient study. So here we have the option to be collaborating with one of our researchers that's testing a CAR T therapy. We have 14 patients, all with different forms of B-cell malignancies. They've been treated with CAR T CD19, and we get their blood at these different time points after they've been infused with the treatment. And this way we're going to be able to look at a really large cohort of samples throughout this study and try to understand where are we seeing like a gene expression signal for patients that have experienced toxicity, which is a problem sometimes after CAR T. So you can see we get these blood, then they are also taking some other samples, they're bringing them to flow, and so this is a big collaboration where we're going to be able to like compare a bunch of the data. There's a lot of processing here in the same way we used to complain about pipetting with some of these plate-based assays. Now here we are again, and this is why I think people will be thinking about automation, whether it's Integra or some other liquid handling system, because you do have to understand like how often you could actually do this assay in a week and what does that workflow look like. What's great here is the blood gets thrown in the freezer, and then it goes through some processing through these stem cell magnets and beads where we isolated leukocytes. Here's my awesome lab tech in there. Because we did this in tubes, that's the way this works, as opposed to being able to sometimes also fix cells in plates. This was a little bit more like labor intensive, where we kind of worked together, because for me about sample prep, counting is a big thing. We wanted mixing. So we knew, like, we had counted. We're lucky we have a selenome. I think this is the big time constraint too. So we were able to like count 24 samples at a time. So we had to do that 4 times, create the sheet to figure out how many cells to put in the plate. And so we did this as like a tag team. We're mixing volume, handoff. We basically ran it through the assay with these, basically the probes now are just probes, and then you have a sample barcode like everyone's described, only we threw in our custom set of probes that we had designed a long time ago for our earlier Visium pilot project, and here you just basically kind of redesign the end so that it works for this assay, and cross your fingers, but it is a little stressful because you take all of these samples through these plates, and then somehow you end up with just 4 tubes with this clear liquid because you've split all of this into 4 gems and you're getting 1 million samples per gem. And you just pray that somehow this is all gonna work out, right? And you're gonna do a lot of sequencing. And actually, we just got some preliminary data on this, so it's still a bit in the data analysis process, but we looked at just 1 gem, so this is 1 million cells, or close to 1 million cells. We're hitting our target, and we had to do some forced cell calling because this was leukocytes. But we see really great metrics so far. You get these printouts that kind of show you where you think your cell per sample expectation is. There's some variability here, but what we were most excited about is like this just came off the computer on Thursday before I came, or Wednesday. And what you can see on the top is like the UMAP plot of where we're seeing these WPRE, which is our custom probe for the CAR Ts, is showing up in this like kind of different sets of clusters. And if I look on the bottom, which is a little bit hard to see, in the pink is where we like do not want to see this signal. So you can kind of point out, I wish I could see the pointer here. I'll just say, we know now that like we're not seeing them here where we see them up here, and like the gray here is like day 0. So like we wouldn't be expecting them to see them. Like we basically don't want to see them where there's pink on top. And so far that looks good. Moving on to the next study, we have fixed PBMCs, only we're treating this in like a consortium like kind of pilot. So imagine you're working with researchers like, say, all over the world. I want to, have Catharine prep some cells. She fixes them, throws them in a dry ice, ships them to me, and you do the same. And, we're all kind of working on a similar question. In this case, we tried this out with a bunch of researchers with their cell of choice. So basically, we let them, they had to be PBMCs, whatever they were interested in. We had a wide variety of cells, from women's estrus cycle to, treated tumor samples, and every lab also had to fix our healthy donor sample. So that was like the internal control. We were fixing them, and we wanted to compare how their processing looked compared to our samples. And so I kind of coordinated all of this with all these labs across the center, and then they brought them in, and then we ran it. And this is even more recent data because this is just based on a shallow sequencing run, because we kind of do a little QC before really committing to that NovaSeq run. And we're getting like pretty good expectation of like what we were hoping to see as far as the number of cells, just a little under 1 million. What we're seeing here in the knee plot is I have 9 samples. Like they were talking about, I knew these were bad. Like I knew these were bad because he told me that I made them report all their viabilities, but these were the ones that supposedly could have this custom probe in it. And so these basically got pooled as if they were like the numbers of everybody else's cells, but there's fewer of them because the viability was lower. Now, what do you do there? Do you like load more? Do you try to make up for that difference? Those are questions that you need to have with your researchers. And so we're really excited to kind of dig a little bit more into this data, obviously, when I get back. Finally, the third one is we just got these blocks. So just like Joe was talking about, we now have this opportunity, and we're really excited about being able to kind of get into those like stores of FFPE blocks from pathologists. So in this study, we're collaborating with Andres Hexy, who had a study earlier on hepatocellular carcinoma, where they looked at 24 samples of, or 24 patients that were treated with, I want to say it's this interleukin 15 CAR T cells, right? And they saw these really different responses. And so what we're doing is we've just gotten these blocks. There's 80 of them, and they're all patients that have had this carcinoma. And what we want to do using this method is to be able to create a tumor landscape to better understand in the future patients that are treated kind of what that response looks like compared to the different types of tumors that they've collected historically, and maybe help them either develop something different or just basically understand why different patients are responding differently. And so this one we're just getting started on. It is true, this heat-based deparaffinization method is super exciting, and we're working on that now. I'm a big believer in practice first, and so we are testing with some blocks. How does this look to do this in the plate? Are we getting the recovery that we think we are? It says 2 scrolls. I have big blocks. What if the sample's smaller? How many cells do I get back? What does this look like if I put it in a tube and compare the number that I get back from the plate? Because all these samples vary, their cell densities vary, and you really are hoping to kind of streamline this by being able to start with the same number of cells so you're not having to do so many counting steps. But if you are going to do this in the plate and you're kind of relying on that as being part of the streamlining, but all your cells are still different concentrations or numbers, then you're still kind of cut with this counting and transferring. So we're testing that before we get started. And how am I doing on time? I'm almost out. Oh my goodness. All right. Well, I'm going to throw up just a few other examples of some benchmarking studies that we're doing, because we also work in the spatial realm. And so we do a lot of testing between spatial platforms with a single set of samples. And this isn't always your perfect, I did this study all at once and have consecutive sections, because we keep coming back to things as they're developed. But we do look at, again, for us, the biology, how easy is it for us to resolve T cells? And obviously when you have these bigger panels, there's less sensitivity, so sometimes that's not the best choice for looking at certain types of groups of cells you want. But we did find that in some cases, like with the cosmics and xenium comparison, that we were having a harder time distinguishing these populations due to some background expression or the higher false discovery rate. And this has been shown, through banana plots. Another one that we've been doing, and Giannis has got this system, is out of Singular. We like the idea of this. There's some different sample processing, but we're also comparing it to some xenium runs. We always have these tissues that we've done on multiple platforms. They just did a recent chemistry upgrade. In the first round, it didn't look so great, but this V2 chemistry is definitely looking to do a better job at resolving these clusters. So this was data for our RCC samples. And then for our TMA, what's going on there? For our TMA, this is like work in progress, because we're just getting into this idea of multimodal spatial data comparison. So we're recognizing that there's some serious differences about how the platforms are actually processing the data and how you take that into consideration. So whether you're normalizing, scaling, we see here that there's a big difference in the protein intensity for the Singular versus xenium data, but we don't think that that necessarily is meaningful at this stage until we really start kind of picking this apart. And it also is informed by kind of how we're doing our cell segmentation and what that means for where we're seeing the protein expression. And so that's it. I mean, basically, yeah, benchmarking is going to keep happening. I think it's valuable. And still choosing a platform is hard. I love that you come up with tools and you get to work with your customers and have those conversations and try and find the right thing for them based on their question or cost or whatever reason. I think we're going to have a lot of new challenges and some fun times trying to figure out how to get through some of these higher throughput methods. And I look forward to kind of hearing input and working with folks on navigating multimodal data integration. So thank you so much for your time. Oh, I forgot my acknowledgement slide.
All right, if you have questions, please come up to the mic and ask away.
So, thanks so much for doing all this fantastic benchmarking and for sharing it with us. So sometimes PIs come to us with FFPE samples, and Flex has been a really nice tool for us. But when we're looking at the nuclei, we're always going to count the nuclei. The problem is that sometimes they've been so aggressively fixed that it degrades the permeability of the nuclear membrane, or it seizes the transcripts up to a point where they're not going to get any signal. And the problem is they won't know that until they get to the end of the bioinformatics, and then they yell at us. And I'm just wondering, is there any way that we could have them yell at you?
They always do.
All right. Are these on? Maybe. Possibly. Okay, cool. Awesome. Yeah, so what we typically do, especially with nuclei, is we do require that they do work with the microscopy core, as I kind of mentioned, just to make sure that like we're not seeing any beveling, that the nuclei is looking really nice. And if we see anything from our end that looks questionable, there's a few things that we kind of like want to put into place. We always like contact our FAS. They see these type of samples coming through all the time, especially like we're pretty, we bug our 10x FAS a lot just to make sure that we're on the right path. But that also kind of adds a nice little sanity check that even if we are telling them these do not look good, maybe don't move forward. If we have the company also saying these do not look good, proceed at your own risk, that could be another conversation just to kind of mitigate that.
Hi, I'm Jen. I work at the Whitehead Genome Core. I have two questions, mostly for 10x or 10x users. The first is, I'm wondering, obviously live cells is, as you guys explained, is a real problem, scheduling, coordinating with people to bring live cells for GEMX preps. And we did have someone try it with DSP fixed cells and said they got decent data. But I'm wondering if anybody else has worked with putting fixed cells directly into GEMS, DSP fixed cells. And I can ask my second question after, if you want.
No, go for it.
Oh, okay. The other one is about Flex V2. We have human and mouse transcriptome probe sets available commercially, and people are designing their own custom probe sets. But I'm wondering, is there, like if we have a Drosophila project and we need a probe set, is there a repository somewhere, like a shared resource? Are we on our own to design those? Is 10x ever going to create a catalog of organisms? Those are my two questions. Thanks.
Well, I'll start for the DSP part. We did a lot of testing between Fresh and basically they showed this comparison in the method document, and so we said again, I don't know, is this work? And so we tried that ourselves with just the PBMCs, and then we actually tested something else because they said that you should, I don't remember if it's like fix and then freeze and then you could go sort, and you're supposed to do your sorting and then fix them and then freeze them, and we thought, well, we also want the flexibility of the sorter, so we did an experiment where we fixed and we had antibodies that like would work for being able to do this fixation, and then we froze those and then we went to the sorter. So we really are very like big on the idea that, like you said, not being there till 9 o'clock at night and people being able to fix their samples. But that other study that I said out of Duke, we didn't get to do a lot of the complete DSP comparisons, but they did. So that would be something I would check out as well.
Duke?
Yeah, the study that they did, I can share that with you.
Okay.
But who else has done DSP? I think it's great.
Not yet.
Not yet?
Nobody's done DSP
Oh.
on GEMS.
Yeah, I've done it a lot on GEMX.
But we still need to wash.
Yeah, you have some loss. It depends on your sample, like the number of cells being input, because you will still have some loss steps.
But in theory, you can just take DSP fixed cells and load them right in without having to reverse the fixation.
Well, you still have to do the decross.
Because that's the test that I did, was someone said,
Yeah.
don't unfix them, just load them. And it didn't, and they
In the past, we have done methanol fixed samples, and that wasn't a success for us. Anybody from the audience who has done DSP fixed? Or any kind of fixing?
But you had to rehydrate it.
Yeah, that's
Yeah. Okay.
yeah. When I'm saying DSP, I mean the DSP methanol fixation, like the one that's part of their protocol, and that you can then freeze.
Okay.
But you do have to bring them out and kind of
And
wash
what
them.
about the whole transcriptome probes?
Yeah,
Does anybody
I
have
was going
any-
to comment on that. So I know from what I understand internally, we have RAT coming, and then NHP should be coming a little bit after that as well. So that can be anticipated. On your question of like a Drosophila one, it's a common question, and I don't think we have like, again, maybe I can leave it up to Ben behind you over there too. But the answer, is kind of it's hard to know which is the best sets to make, right? And so maybe a repository resource would be helpful, but that's just something I can't comment on like where we're at on making that type of resource. But I know kind of coming down the line, we should have additional species available that should kind of add to the number of species you can access.
Yeah,
as Flex.
I don't know if this is something that people who design these probe sets like to protect and keep to themselves, or if there's a mission to make a resource for people.
I love that idea.
Yeah, it's
I mean,
great.
especially at Star Wars, we work with all these weird organisms, and it feels like every time it's a process. And we have the PI lab work with the bioinformatics team to design these probes, but I'm sure when they publish, they'll make it available, but
Right.
there's no resource really. So like a vendor like Twist should probably take this on and consider creating like a public space for us to do this kind of thing.
Thank
Yeah,
you.
Great point.
Love the test and practice before implementing benchmarking. I kind of curious on two things. One, is it something you've kind of built into your funding to kind of test ahead of time, or are you go the model of the investigators pay like a heavy discount? It's like a scratch my back, scratch yours kind of agreement for the kind of pre-testing that y'all do?
Yeah, well, my funding is like, I didn't get to that slide. My funding is a little bit different. It's kind of an internal within our immunotherapy group. And so because of the way our lab was designed, I'm not a true like fee-for-service lab. Like if they run something on an instrument, I might have a fee to help cover a service contract cost. But this is really kind of meant for us to be sort of like this core for this research group. And for us, we kind of almost joke a little bit that we're like, we can fail so you don't have to. And the idea is to kind of minimize cost across all of these different research groups having to, buy something, test it, fail with it, different instruments across the place. And so if we like something and we will then try to move it towards the core and kind of like maybe worked out some of the kinks, they also will bring on and test their own stuff. But our funding is a little bit separate than a traditional core.
Okay, so people will go to you with the weird stuff that may not
People
that's
go to-
not going to work, and you make it work.
Yeah, and we had a great example with a nuclei sample that should have been like simple and forward frozen colon tissue from a mouse, and we just put out a STAR protocol because whatever it said in the 10x thing to do this didn't really work well for that sample, and so we went a little above and beyond and tried out some reagents from Miltenyi. And they got chillers. Like you'd go check out their booth down there, and like the data was beautiful. I don't know why it just made the difference in that case.
And then I love the reducing the low sequencing depth pre-check. How low can you go?
Yeah, I don't know exactly. I mean, in this case, someone was talking yesterday at Illumina, like the workhorse. Like, so we have a genomics core that we send all the NovaSeq stuff out to, but we have a NextSeq downstairs, which we have access to and we can load. And so we just that's why we chose the one gem. And I think I got about 500 reads per cell or something. The Eric's get about 100 million. And we got some results that at least indicate that nothing's horrible. But I don't think I would go much lower than that.
Decent concordance, 500.
Yeah.
Okay,
Yeah.
good to hear.
What about you, Kat?
You probably have a
We
lot
do
more.
it on the I-100, so it's
Yeah.
faster.
Yeah, I was thinking of ways to utilize I-100 more.
Yeah.
Yeah.
So
But I don't know
we
if
do
I'd
that.
put 1 million cells on the I-100.
Yeah, we
Yeah.
just use the smallest one and then see.
Yeah.
So it's mostly like to see the complexity,
Yeah.
the barcodes and so.
What's-
About, about your budget question, so we have both scenarios where the core has a development budget, and we use that to do some testing. But if you have like a PI who wants to do some kind of crazy experiment, we have them do like 50-50 with the core.
Okay,
Yep.
that's a good model.
I know we are a minute over time. I would say for the vendors in this room, something to consider is there are a lot of groups getting into bacterial single-cell sequencing. So please, please, please try to come up with like something that we can use.
Yeah, that was a good session yesterday.
All right.
Yeah.
Thanks a lot. Thanks for being here.